In the quiet hours, when the world slows and the rhythms of life settle into a gentle hum, there is another rhythm at work, one far too small to see, yet endlessly intricate. It is the rhythm of chemistry inside living things, a ceaseless dance of molecules, of atoms joining and parting, of delicate exchanges that have been unfolding for billions of years. This is the world of biochemistry, the study of life at the molecular level. It is a science that listens closely to the whispers of the smallest components of existence, finding meaning in the way they meet, interact, and transform. Every living cell, no matter how tiny or complex, is a miniature chemical world. Within its boundaries, thousands of reactions take place in perfect coordination. Sugars are broken down for energy. Proteins are folded with care. Signals pass softly from one molecule to the next, like messages passed in a great silent hall. And all of it, every intricate movement, follows the laws of chemistry. Biochemistry is a bridge between biology, the study of life, and chemistry, the study of matter. It is where the two meet and speak the same language. Here, life is not seen as a mystery to be solved suddenly, but as a long unfolding pattern where each answer leads softly to the next. This pattern connects all living things, from the green leaf drawing in sunlight to the fish gliding through a stream, to the quiet breath rising and falling in your chest. Though the details differ, the molecules are the same. The energy flows in the same gentle currents. The chemistry is universal. Even the most ordinary moments, the slow beat of your heart, the way your body processes the food you ate hours ago, the simple act of breathing in and out, are guided by a hidden network of biochemical reactions. In this way, biochemistry is both invisible and everywhere. It can feel remarkable to think that you are, in a sense, a collection of carefully arranged molecules. Yet that arrangement is not random. It is purposeful, precise, and sustained by a constant exchange of matter and energy. Inside you, a single heartbeat is accompanied by countless chemical changes. Molecules of glucose are broken apart to release energy, a process that has been occurring since the earliest life forms first emerged on Earth. The very same oxygen you breathe in travels through the bloodstream and slips into your cells, where it helps to complete this ancient process. The food you eat, whether fruit, grain, or protein, is not simply absorbed whole. Instead, it is taken apart in slow, careful steps. Large molecules are broken into smaller ones, then reassembled into forms your body can use. Each transformation is guided by enzymes, the gentle catalysts of biochemistry, who nudge reactions forward without forcing them. Water, too, is everywhere in this microscopic world. It is the silent stage on which all biochemical reactions occur. Every cell is a tiny chamber of water filled with dissolved salts, sugars, and proteins. Without water's remarkable properties, its ability to dissolve so many substances, its capacity to absorb heat without changing temperature quickly, the delicate balance of life's chemistry would collapse. Water molecules are constantly in motion, flowing into and out of cells, shaping their volume, carrying nutrients to where they are needed, and removing waste when it is no longer useful. This movement is so ordinary, so constant, that it is easy to forget it is happening at all. But without it, life could not persist for even a moment. If you were able to shrink down to the scale of a single molecule, the world would appear very different. You would see a crowded, dynamic space. Proteins would fold into complex shapes beside you. Lipid molecules would drift together, forming the flexible membranes that enclose each cell. Tiny strands of nucleic acids would curl and twist, carrying coded instructions written in a molecular language older than any human tongue. There would be no empty silence here, not because it is noisy, but because it is busy. Reactions would be taking place in every direction, yet not chaotically. Each has its place, its timing, and its purpose. The cell is like a city with no wasted movement. Food arrives just where it is needed. Messages are sent without delay. Waste is carried away efficiently. And while the city of the cell is active, it is also harmonious. One process flows into the next. The breakdown of one molecule provides the starting material for another. Energy produced in one reaction is stored, then drawn upon by another, keeping the cycle unbroken. Biochemistry is the study of this harmony. It asks what molecules are present in living things. How do they interact? What patterns do they follow? And perhaps most beautifully, how do such patterns give rise to life itself? This is not the chemistry of explosions or sudden change. It is the chemistry of steady, ongoing transformation. A chemistry that unfolds in whispers, shaping everything we know as life. It is comforting to think that these same molecular processes are not unique to us. The chemical pathways in a tree leaf, in a hummingbird's wing, in the roots of grass, they are variations on the same themes that flow through our own cells. The glucose molecule that fuels a cell in your body is built in much the same way as the glucose molecule made in a green leaf by sunlight. The ATP molecule that carries energy in a bird's muscle is identical to the one in your own. This shared chemistry is a reminder that all living things, no matter how different they seem, are bound together by the same deep currents. As we begin this quiet exploration of biochemistry, we will see that these currents follow certain rules. They are shaped by the properties of atoms, by the bonds they form, and by the environments in which they move. We will see how small molecules join to create larger ones, and how these larger molecules fold, twist, and interact to form the machinery of life. We will learn how energy moves within a cell, how it is stored, and how it is used. We will look at the delicate balance between building up and breaking down, the endless cycle of creation and transformation that sustains us. We will see how cells send messages, how they sense their surroundings, and how they respond to subtle changes in their internal chemistry. And we will reflect on how all of this, from the simplest chemical reaction to the most complex genetic process, is part of one vast and continuous pattern. So as we set out, it may be helpful to imagine biochemistry not as a subject to be hurried through, but as a landscape to move through slowly. A landscape where every molecule has a story and every reaction is part of a greater whole. In this landscape, the pace is never frantic. Even the fastest biochemical reactions occur in the context of a life that is measured not in seconds alone, but in seasons, generations, and eons. These processes have been in motion since long before humans appeared, and they will continue long after. So breathe slowly, let your mind settle in the thought that even now, while you are still, your cells are quietly alive with activity, energy is flowing, molecules are moving, and the chemistry of life carries on, always and without pause. This is where our journey begins, in the smallest spaces, with the quietest motions, in the delicate, constant chemistry of life. At its heart, biochemistry is the study of the molecules that make up living organisms and the reactions these molecules undergo. It sits at the intersection of biology and chemistry, examining how the principles of chemistry apply to biological systems. While chemistry can describe reactions in a test tube, biochemistry asks how those same reactions occur within the confines of a cell, governed by the unique conditions of life. It investigates not just what molecules are present, but how they interact in coordinated ways to produce the phenomena we recognize as living. The scope of biochemistry is vast. It encompasses the structure and function of biomolecules like carbohydrates, lipids, proteins, and nucleic acids. It explores metabolism, the sum of all chemical reactions occurring in an organism. It examines how genetic information is stored, transmitted, and expressed. It considers how cells communicate with one another through signaling molecules. And it investigates how organisms respond to their environment at the molecular level. In essence, biochemistry seeks to understand life through the lens of molecular interactions. To appreciate biochemistry, we begin with the atom. Atoms are the fundamental units of matter, the building blocks from which everything is constructed. Each atom consists of a nucleus containing protons and neutrons, surrounded by a cloud of electrons. The number of protons defines the element. Carbon has 6 protons, nitrogen has 7, oxygen has 8. These 3 elements, along with hydrogen, which has just 1 proton, make up the majority of the mass of living organisms. Together, carbon, hydrogen, nitrogen, and oxygen form the backbone of biochemistry. Carbon is especially important. It has 4 electrons in its outer shell, which allows it to form 4 bonds with other atoms. This gives carbon remarkable versatility. It can bond to other carbon atoms, forming long chains and rings, creating the structural diversity necessary for complex molecules. Carbon's ability to form stable bonds with hydrogen, oxygen, nitrogen, sulfur, and phosphorus allows for the construction of the vast array of organic molecules found in living things. It is no exaggeration to say that carbon is the element of life. Atoms bond together by sharing or transferring electrons. A covalent bond forms when 2 atoms share a pair of electrons. These bonds are strong and stable, forming the framework of most biological molecules. In a molecule of water, for example, each hydrogen atom shares an electron with the central oxygen atom, creating 2 covalent bonds. The result is a small, simple molecule with profound importance. A single covalent bond, represented by a line between 2 atoms, is the fundamental connection that holds molecules together. Some covalent bonds are polar, meaning the electrons are not shared equally. Oxygen, being more electronegative than hydrogen, pulls the shared electrons closer to itself in a water molecule. This creates a slight negative charge near the oxygen and slight positive charges near the hydrogens. Water is therefore a polar molecule, and this polarity gives it many of its unique properties. Polar molecules interact readily with other polar molecules, while nonpolar molecules, like oils, do not dissolve well in water. This simple principle underlies much of biochemistry, influencing how molecules behave in the aqueous environment of the cell. Hydrogen bonds are another type of interaction, weaker than covalent bonds, but incredibly important. A hydrogen bond forms when a hydrogen atom covalently bonded to an electronegative atom, like oxygen or nitrogen, is attracted to another electronegative atom nearby. In water, each molecule can form hydrogen bonds with up to 4 neighboring water molecules, creating a dynamic, interconnected network. Hydrogen bonds are constantly forming and breaking, giving water its fluidity while also providing cohesion. These bonds also stabilize the structures of proteins and nucleic acids, holding complex shapes in place without the rigidity of covalent bonds. Ionic bonds result from the transfer of electrons from one atom to another. When sodium loses an electron, it becomes a positively charged ion. When chlorine gains an electron, it becomes a negatively charged ion. The attraction between opposite charges holds them together. In the dry crystal of table salt, ionic bonds are strong, but in water they dissolve easily as water molecules surround and stabilize the separated ions. Inside cells, ions like sodium, potassium, calcium, and chloride play crucial roles in signaling, maintaining electrical gradients, and regulating cellular functions. At the molecular level, life depends on these interactions: covalent bonds for structure, hydrogen bonds for flexibility and specificity, ionic interactions for charge-based signaling. Together, they allow molecules to come together, perform functions, and then dissociate when their work is done. This dynamic quality is central to biochemistry. Molecules are not static structures locked in place. They are constantly interacting, changing partners, responding to their environment. It is a molecular dance, choreographed by the laws of chemistry and shaped by the needs of life. Water is often called the solvent of life, and with good reason. It makes up about 60 to 70% of the mass of most living organisms. Inside cells, water is the medium in which biochemical reactions occur. Its polarity allows it to dissolve a wide range of substances, from salts to sugars to proteins. When a salt crystal dissolves in water, water molecules surround each ion, forming a hydration shell that stabilizes the ion in solution. This ability to dissolve so many substances makes water an ideal transport medium, carrying nutrients, gases, and waste products throughout the body. Water also has a high specific heat, meaning it can absorb or release large amounts of heat without changing temperature dramatically. This property helps organisms maintain stable internal temperatures despite fluctuations in the external environment. The hydrogen bonds between water molecules require significant energy to break, so water resists temperature changes. This thermal stability is crucial for maintaining the conditions necessary for biochemical reactions, which often proceed optimally within narrow temperature ranges. Water's cohesion, the tendency of water molecules to stick together, creates surface tension and allows water to move through narrow spaces, such as the vessels in plants or the capillaries in animals. Its adhesion, the tendency to stick to other surfaces, works alongside cohesion to enable this movement. Water also expands when it freezes, becoming less dense as ice, which is why ice floats. This seemingly simple fact has profound ecological implications, allowing aquatic life to survive beneath frozen surfaces. In biochemistry, water participates directly in many reactions. Hydrolysis reactions break bonds by adding water, splitting larger molecules into smaller ones. Condensation reactions, also called dehydration synthesis, join molecules together by removing water. These reactions are fundamental to building and breaking down the macromolecules of life. Water is not merely a passive backdrop. It is an active participant shaping the structure and reactivity of biomolecules. Living organisms are composed primarily of four major classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Each class has distinct structures and functions, yet all are built from smaller repeating units. Carbohydrates are composed of sugars. Lipids are largely hydrophobic molecules. often containing fatty acids. Proteins are polymers of amino acids. Nucleic acids are polymers of nucleotides. Understanding these macromolecules is central to understanding biochemistry, for they are the molecules that carry out the work of life. Carbohydrates serve primarily as sources of energy and as structural materials. The simplest carbohydrates are monosaccharides, single sugar molecules like glucose, fructose, and galactose. Glucose, a 6-carbon sugar, is perhaps the most important monosaccharide, serving as the primary fuel for cellular respiration. Its structure is a ring of 5 carbons and 1 oxygen with hydroxyl groups attached. This simple molecule is the starting point for energy production in nearly all living cells. When 2 monosaccharides join together, they form a disaccharide. Sucrose, common table sugar, is a disaccharide made from glucose and fructose. Lactose, found in milk, is made from glucose and galactose. Maltose, formed during the digestion of starch, consists of 2 glucose molecules. These disaccharides are easily broken down into their component monosaccharides, which can then be used for energy. Polysaccharides are long chains of monosaccharides, sometimes branching, sometimes linear. Starch, found in plants, is a storage form of glucose. It consists of long chains of glucose molecules linked together, coiled into granules inside plant cells. When an organism consumes starch, enzymes break the bonds between glucose units, releasing individual glucose molecules that can be absorbed and used. Glycogen is the animal equivalent of starch, a highly branched polymer of glucose, stored primarily in the liver and muscles. When energy is needed, glycogen is broken down to release glucose into the bloodstream. Cellulose is another polysaccharide made of glucose, but its bonds are arranged differently, creating long, straight chains that pack tightly together, forming fibers of great strength. Cellulose is the main structural component of plant cell walls. Humans cannot digest cellulose because we lack the enzyme needed to break its specific bonds, but it serves as dietary fiber, aiding digestion. Other organisms, like cows and termites, harbor bacteria in their digestive systems that can break down cellulose, allowing them to extract energy from plant material that would otherwise be inaccessible. Carbohydrates are not only fuels and structures, they also play roles in cell recognition and signaling. Short chains of sugars attached to proteins or lipids on the surface of cells act as identification tags, helping cells recognize each other and communicate. This is especially important in the immune system, where cells distinguish between self and non-self-based, partly on surface carbohydrates. Lipids are a diverse group of hydrophobic molecules. Unlike carbohydrates, proteins, and nucleic acids, lipids are not polymers. They do not consist of repeating units. Instead, they are defined by their solubility. Lipids are largely insoluble in water, but dissolve readily in non-polar solvents. This hydrophobic character allows lipids to form structures like cell membranes and to serve as concentrated energy storage. The most common lipids are fats and oils, technically called triacyl glycerols or triglycerides. A triglyceride consists of a glycerol molecule, a 3-carbon backbone, bonded to 3 fatty acids. Fatty acids are long hydrocarbon chains with a carboxyl group at one end. The hydrocarbon tail is hydrophobic, while the carboxyl group is hydrophilic. When 3 fatty acids attach to glycerol, the result is a highly hydrophobic molecule that aggregates with other lipids, forming droplets inside cells. Fatty acids vary in length and in the number of double bonds they contain. Saturated fatty acids have no double bonds. Their hydrocarbon chains are straight, allowing them to pack tightly together, which is why saturated fats tend to be solid at room temperature, like butter. Unsaturated fatty acids contain one or more double bonds, which introduce kinks in the chain. These kinks prevent tight packing, so unsaturated fats tend to be liquid at room temperature, like olive oil. The degree of saturation affects not only physical properties, but also biological functions. Phospholipids are similar to triglycerides, but have only two fatty acids. The third position on the glycerol is occupied by a phosphate group, often with an additional chemical group attached. This creates a molecule with a hydrophilic head and two hydrophobic tails. In water, phospholipids spontaneously arrange themselves into bilayers, with the hydrophobic tails facing inward, away from water, and the hydrophilic heads facing outward toward the aqueous environment. This bilayer forms the foundation of all cell membranes, creating a barrier that separates the inside of the cell from the outside while remaining flexible and fluid. Steroids are another class of lipids characterized by a structure of 4 interconnected carbon rings. Cholesterol is the most well-known steroid, a component of animal cell membranes that helps regulate membrane fluidity. Cholesterol also serves as a precursor for the synthesis of steroid hormones like testosterone, estrogen, and cortisol, which regulate a wide range of physiological processes. Though often vilified in discussions of health, cholesterol is essential to life. Lipids store more than twice as much energy per gram as carbohydrates, making them an efficient form of long-term energy storage. Adipose tissue, made up of cells filled with triglycerides, serves as an energy reserve that can be drawn upon when food is scarce. Lipids also provide insulation and cushioning, protecting organs and helping maintain body temperature. Beyond these roles, certain lipids function as signaling molecules, vitamins, and pigments, highlighting the versatility of this class of biomolecules. Proteins are perhaps the most functionally diverse macromolecules. They catalyze reactions, transport molecules, provide structure, defend against pathogens, regulate gene expression, and enable movement. Nearly every process in the cell involves proteins in some capacity. This functional diversity arises from the vast variety of protein structures, which in turn arise from the sequence of amino acids that make up each protein. An amino acid is a small molecule with a central carbon atom bonded to 4 groups: an amino group, a carboxyl group, a hydrogen atom, and a variable side chain called the R group. The R group differs among the 20 standard amino acids, giving each its unique properties. Some side chains are hydrophobic, others are hydrophilic. Some are acidic, some are basic, some are small, some are large. This diversity allows proteins to adopt a wide range of shapes and perform a wide range of functions. Amino acids link together through peptide bonds, covalent bonds formed between the carboxyl group of one amino acid and the amino group of another, releasing a molecule of water in the process. A chain of amino acids is called a polypeptide. When a polypeptide folds into a functional three-dimensional shape, it becomes a protein. The sequence of amino acids, determined by genetic instructions, dictates how the polypeptide will fold and what function it will perform. The structure of a protein can be described at 4 levels. The primary structure is simply the linear sequence of amino acids. The secondary structure refers to local folding patterns stabilized by hydrogen bonds, such as alpha helices, spiral shapes, and beta sheets, accordion-like structures. The tertiary structure is the overall three-dimensional shape of a single polypeptide formed by interactions among side chains, including hydrogen bonds, ionic bonds, disulfide bridges between cysteine residues, and hydrophobic interactions. The quaternary structure applies to proteins made of multiple polypeptides, describing how those subunits fit together. The shape of a protein is crucial to its function. Enzymes, for example, have specific active sites, pockets, or grooves where substrate molecules bind. The shape of the active site determines which substrates can bind and which reactions can be catalyzed. If a protein loses its shape through heat, changes in pH, or chemical disruption, it becomes denatured and loses its function. This underscores a central principle of biochemistry: structure determines function. The precise arrangement of atoms in a protein enables it to carry out its specific role in the cell. Enzymes are biological catalysts, proteins that speed up chemical reactions without being consumed in the process. Life depends on enzymes. Without them, most biochemical reactions would proceed far too slowly to sustain life. Enzymes work by lowering the activation energy of a reaction, the energy barrier that reactants overcome to become products. By stabilizing the transition state, the high-energy intermediate form of the reactants, enzymes make it easier for reactions to proceed. Each enzyme is specific to a particular reaction or type of reaction. This specificity arises from the shape of the enzyme's active site, which is complementary to the shape of its substrate. When a substrate binds to the active site, the enzyme undergoes a slight change in shape, an induced fit, that brings reactive groups into optimal positions. The reaction occurs, products are released, and the enzyme returns to its original state, ready to catalyze another reaction. Enzyme activity can be regulated in several ways. Some enzymes are controlled by the binding of regulatory molecules to sites other than the active site, a process called allosteric regulation. Binding of an activator enhances enzyme activity, while binding of an inhibitor reduces it. This allows cells to fine-tune metabolic pathways in response to changing conditions. Enzymes can also be regulated by covalent modification, such as the addition or removal of phosphate groups, which can activate or deactivate the enzyme. Feedback inhibition is another common regulatory mechanism where the product of a pathway inhibits an enzyme earlier in the pathway, preventing overproduction. Nucleic acids, DNA, and RNA are the molecules of heredity and gene expression. They store and transmit genetic information, the instructions for building and operating an organism. Like proteins, nucleic acids are polymers, but instead of amino acids, they are made of nucleotides. Each nucleotide consists of 3 components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. In DNA, the sugar is deoxyribose, and there are 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. In RNA, the sugar is ribose, and uracil replaces thymine. Nucleotides link together through bonds between the sugar of one nucleotide and the phosphate of the next, forming a sugar-phosphate backbone with the bases projecting outward. DNA typically exists as a double helix, 2 strands wound around each other with the bases on each strand pairing in a specific way: adenine with thymine and guanine with cytosine. These base pairs are held together by hydrogen bonds. The sequence of bases along a DNA strand encodes genetic information. Genes are segments of DNA that specify the sequence of amino acids in a protein. The process begins with transcription, where a segment of DNA is copied into a molecule of messenger RNA. This mRNA then serves as a template for translation. where ribosomes read the sequence and assemble amino acids into a polypeptide. The flow of genetic information from DNA to RNA to protein is sometimes called the central dogma of molecular biology. RNA has several forms, each with distinct functions. Messenger RNA carries genetic information from DNA to the ribosome. Transfer RNA brings amino acids to the ribosome during protein synthesis. Ribosomal RNA, along with proteins, makes up the ribosome itself. Other RNA molecules play roles in regulating gene expression and processing other RNAs. Together, these nucleic acids coordinate the expression of genetic information, ensuring that the right proteins are made at the right times in the right amounts. We have walked through the foundation, exploring the atoms, bonds, and molecules that form life's alphabet. Now we continue by observing how this alphabet creates words, sentences, and eventually entire stories. We turn our attention to metabolism, the sum of all chemical reactions occurring in an organism. Metabolism is not a single process, but a network of interconnected pathways, each one a series of enzyme-catalyzed reactions that transform molecules step by step. Metabolism can be divided into two broad categories. Catabolism refers to the breakdown of complex molecules into simpler ones, releasing energy in the process. Anabolism refers to the synthesis of complex molecules from simpler ones, requiring energy input. These two processes are complementary. The energy released by catabolic reactions is captured and used to drive anabolic reactions. This coupling of energy-releasing and energy-requiring processes is central to how cells manage energy. Energy in biological systems is carried primarily by a molecule called ATP, adenosine triphosphate. ATP consists of adenine, ribose, and 3 phosphate groups. The bonds between the phosphate groups are high-energy bonds, meaning their hydrolysis releases a significant amount of energy. When ATP is hydrolyzed to ADP, adenosine diphosphate, and an inorganic phosphate, energy is released and can be used to drive other reactions. ATP is often described as the energy currency of the cell, constantly being spent and regenerated. Cells produce ATP primarily through cellular respiration, a series of catabolic pathways that extract energy from glucose and other organic molecules. Cellular respiration occurs in 3 main stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. Together, these pathways oxidize glucose completely, producing carbon dioxide, water, and ATP. The process is efficient, capturing much of the energy stored in glucose and transferring it to ATP. Glycolysis occurs in the cytoplasm and does not require oxygen. It is the first stage of glucose metabolism. A molecule of glucose, a 6-carbon sugar, is split into 2 molecules of pyruvate, each containing 3 carbons. Along the way, a small amount of ATP is produced, and high-energy electrons are transferred to a carrier molecule called NAD+, reducing it to NADH. Glycolysis is an ancient pathway present in nearly all living organisms, suggesting it evolved early in the history of life. The reactions of glycolysis proceed in 10 steps, each catalyzed by a specific enzyme. The pathway begins with an investment phase, where ATP is consumed to phosphorylate glucose and its intermediates. This makes the molecules more reactive and traps them inside the cell. The glucose molecule is then split into 2 3-carbon molecules. In the payoff phase, these molecules are oxidized and energy is captured in the form of ATP and NADH. The net result is a gain of 2 ATP molecules and 2 NADH molecules per glucose. Pyruvate, the end product of glycolysis, stands at a metabolic crossroads. In the presence of oxygen, pyruvate enters the mitochondria and is further oxidized in the citric acid cycle. In the absence of oxygen, pyruvate undergoes fermentation, a process that regenerates NAD+, so glycolysis can continue. Fermentation occurs in some microorganisms and in muscle cells during intense exercise when oxygen supply is insufficient. Lactic acid fermentation produces lactate, while alcoholic fermentation produces ethanol and carbon dioxide. When oxygen is available, pyruvate is transported into the mitochondria, where it is converted to acetyl-CoA, a 2-carbon molecule attached to coenzyme A. This reaction releases carbon dioxide and transfers electrons to NAD+, forming NADH. Acetyl-CoA then enters the citric acid cycle, also known as the Krebs cycle or the tricarboxylic acid cycle. The citric acid cycle is a circular pathway that occurs in the mitochondrial matrix. Acetyl-CoA combines with a 4-carbon molecule, oxaloacetate, to form a 6-carbon molecule, citrate. Through a series of reactions, citrate is gradually oxidized back to oxaloacetate, releasing 2 molecules of carbon dioxide and transferring high-energy electrons to NAD+, and another carrier, FAD. Each turn of the cycle produces 3 NADH, 1 FADH2, and 1 ATP or GTP. Since each glucose yields 2 pyruvate molecules, the cycle turns twice per glucose, doubling these outputs. The citric acid cycle does more than generate energy carriers. Its intermediates serve as starting points for the synthesis of amino acids, nucleotides, and other molecules. This dual role, both catabolic and anabolic, makes the citric acid cycle a central hub of metabolism. Enzymes that regulate the cycle respond to the cell's energy needs, slowing down when ATP is abundant and speeding up when energy is low. The high-energy electrons captured in NADH and FADH2 during glycolysis and the citric acid cycle are not yet in the form of ATP. To convert their energy into ATP, the electrons pass through the electron transport chain, a series of protein complexes embedded in the inner mitochondrial membrane. As electrons move from one complex to the next, energy is released and used to pump protons from the mitochondrial matrix into the intermembrane space. This creates a proton gradient, a difference in proton concentration across the membrane. The flow of protons back into the matrix through an enzyme called ATP synthase drives the synthesis of ATP from ADP and inorganic phosphate. This process, called oxidative phosphorylation, produces the majority of ATP in cellular respiration. At the end of the electron transport chain, electrons are transferred to oxygen, which combines with protons to form water. Oxygen is the final electron acceptor, and without it, the electron transport chain cannot function, and ATP production grinds to a halt. Cellular respiration is remarkably efficient. From one molecule of glucose, the complete oxidation through glycolysis, the citric acid cycle, and oxidative phosphorylation can yield approximately 30 to 32 molecules of ATP, though the exact number varies. This represents about 30 to 40% of the energy in glucose, with the rest released as heat. While not perfect, this efficiency far exceeds that of most human-made engines. The pathways of metabolism do not operate in isolation. They are interconnected, regulated, and responsive to the needs of the cell. Carbohydrates, lipids, and proteins can all be catabolized for energy, and their breakdown products can feed into common pathways. Amino acids, for example, can be deaminated, and their carbon skeletons can enter the citric acid cycle at various points. Fatty acids are broken down through beta-oxidation, producing acetyl-CoA that enters the citric acid cycle. This metabolic flexibility allows organisms to extract energy from a variety of nutrients and to survive periods when certain nutrients are scarce. As we conclude this first part of our journey, we return to where we began. Biochemistry is the study of the molecular foundations of life, the chemistry that occurs within and between cells. We have explored atoms and bonds, water and macromolecules, enzymes, and metabolism. We have seen how glucose is broken down through glycolysis and the citric acid cycle, how electrons are transferred through the electron transport chain, and how ATP is synthesized to power the work of the cell. These are not isolated facts, but threads in a larger tapestry, a continuous pattern of transformation and renewal. Even now, as you rest, these processes continue within you. Glucose is being broken down, ATP is being produced and consumed, proteins are folding, membranes are maintaining their integrity, water is flowing. The chemistry of life is unceasing, a quiet symphony played in the key of molecules, conducted by enzymes and sustained by the energy that flows through all living things. Let this understanding settle gently, knowing more awaits in the pathways yet to explore, in the deeper currents of metabolism that we will walk through together in time. We have walked gently through the foundations, observing how atoms join to form molecules, how water shapes the cellular environment, how macromolecules carry out their diverse functions, and how glucose is broken down to release energy through glycolysis and the citric acid cycle. Now, we continue our journey by exploring how cells capture energy from sources beyond glucose, how they build the molecules they need, and how these pathways interconnect in the living web of metabolism. The processes we are about to explore are not separate from what we have already learned. They are extensions, elaborations of the same fundamental principles. Metabolism is not a collection of isolated pathways, but a network where one reaction feeds into another, where the product of one process becomes the starting material for the next. This interconnectedness is one of biochemistry's most elegant features, revealing how efficiency and flexibility are built into the molecular design of life. Let us begin with photosynthesis, the process by which plants, algae, and certain bacteria capture light energy and convert it into chemical energy stored in glucose. Photosynthesis is the complement to cellular respiration. Where respiration breaks down glucose to release energy, photosynthesis builds glucose by capturing energy. Together, these two processes form a cycle that sustains much of life on Earth. The oxygen you breathe is a byproduct of photosynthesis. The glucose that fuels your cells was likely built by a plant using sunlight. Photosynthesis occurs in two stages, the light-dependent reactions and the light-independent reactions, also known as the Calvin cycle. The light-dependent reactions take place in the thylakoid membranes of chloroplasts, specialized organelles found in plant cells. Here, chlorophyll and other pigments absorb light energy, exciting electrons to higher energy levels. These high-energy electrons are passed through an electron transport chain, similar in concept to the one in mitochondria, though the details differ. As electrons move through the chain, energy is used to pump protons across the thylakoid membrane, creating a proton gradient. The flow of protons back through ATP synthase drives the production of ATP. Meanwhile, the electrons ultimately reduce a molecule called NADP+ to NADPH, a carrier similar to NADH. Water molecules are split in the process, releasing oxygen as a byproduct. This stage of photosynthesis produces ATP and NADPH. the energy and reducing power needed for the next stage. The Calvin cycle occurs in the stroma, the fluid-filled space surrounding the thylakoids. Here, carbon dioxide from the atmosphere is incorporated into organic molecules through a process called carbon fixation. An enzyme called rubisco catalyzes the reaction between carbon dioxide and a 5-carbon sugar, RuBP. The resulting 6-carbon molecule immediately splits into 2 3-carbon molecules. Through a series of reactions powered by ATP and NADPH, these molecules are rearranged and reduced, eventually producing G3P, glyceraldehyde 3-phosphate, a 3-carbon sugar. Some of the G3P molecules leave the cycle and are used to synthesize glucose and other carbohydrates. The rest are used to regenerate RuBP, allowing the cycle to continue. The Calvin cycle turns 6 times to produce 1 molecule of glucose, incorporating 6 molecules of carbon dioxide and consuming 18 ATP and 12 NADPH. It is a process of remarkable efficiency, taking inorganic carbon and transforming it into the organic molecules that form the foundation of the food chain. Photosynthesis connects the energy of the sun to the chemistry of life. The glucose produced by plants is consumed by animals, which break it down through cellular respiration, releasing the stored energy. The carbon dioxide released by respiration is taken up by plants and used in photosynthesis. Oxygen released by photosynthesis is used in respiration. This cycle, moving carbon and energy through ecosystems, has been operating for billions of years, shaping the composition of the atmosphere and enabling the diversity of life we see today. Returning to the pathways within our own cells, let us consider how lipids are broken down for energy. Fatty acids, the building blocks of fats, are rich in energy, containing more than twice as many calories per gram as carbohydrates. When energy is needed and glucose is scarce, the body turns to its lipid stores. Triglycerides in adipose tissue are hydrolyzed, releasing glycerol and fatty acids into the bloodstream. These molecules travel to cells, where they are taken up and metabolized. Glycerol, a 3-carbon molecule, is converted into an intermediate of glycolysis and can be oxidized through the same pathways as glucose. Fatty acids undergo a different process called beta-oxidation. This occurs in the mitochondria and involves the repeated removal of 2 carbon units from the fatty acid chain. Each cycle of beta-oxidation shortens the fatty acid by 2 carbons, producing 1 molecule of acetyl-CoA, 1 NADH, and 1 FADH2. Acetyl-CoA enters the citric acid cycle just as it does after the breakdown of glucose. The NADH and FADH2 donate their electrons to the electron transport chain, driving ATP synthesis. A fatty acid with 16 carbons, such as palmitic acid, undergoes 7 cycles of beta-oxidation, producing 8 acetyl-CoA molecules, 7 NADH, and 7 FADH2. The complete oxidation of a single palmitic acid molecule yields over 100 molecules of ATP, illustrating why fats are such efficient energy stores. Beta-oxidation is regulated according to the cell's energy needs. When ATP levels are high, fatty acid oxidation slows. When ATP is low and glucose is scarce, fatty acid oxidation increases. Hormones like glucagon and epinephrine stimulate the breakdown of triglycerides and promote fatty acid oxidation, while insulin promotes fat storage. This hormonal regulation ensures that lipid metabolism is coordinated with the body's overall metabolic state. Proteins, too, can be catabolized for energy, though they are generally used only when carbohydrates and fats are insufficient. Proteins are first broken down into amino acids. These amino acids can be deaminated, meaning the amino group is removed, leaving behind a carbon skeleton. The amino group is converted to urea and excreted, while the carbon skeleton can enter metabolic pathways at various points, depending on the amino acid. Some amino acids are converted to pyruvate, which can enter the citric acid cycle. Others are converted directly to intermediates of the citric acid cycle, such as alpha-ketoglutarate or succinyl-CoA. Still others are converted to acetyl-CoA. In this way, the carbon skeletons of amino acids can be oxidized for energy, though this represents a loss of valuable building blocks that could have been used to synthesize new proteins. Under normal circumstances, the body preferentially uses carbohydrates and fats for energy, preserving proteins for their structural and functional roles. Metabolism is not only about breaking down molecules. Anabolism, the synthesis of complex molecules from simpler ones, is equally important. Cells continuously build new proteins, nucleic acids, lipids, and carbohydrates to support growth, repair, and reproduction. These biosynthetic pathways require energy, typically provided by ATP, and reducing power, often provided by NADPH. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors. This pathway is essentially the reverse of glycolysis, though it is not simply glycolysis running backward. Several steps in glycolysis are irreversible under cellular conditions, so gluconeogenesis uses different enzymes to bypass these steps. The pathway occurs primarily in the liver and to a lesser extent in the kidneys. Gluconeogenesis is important during fasting or prolonged exercise. When blood glucose levels drop, the body can synthesize glucose from lactate, glycerol, and certain amino acids, ensuring a steady supply of glucose for tissues that depend on it, such as the brain and red blood cells. The regulation of gluconeogenesis is tightly controlled, with hormones like glucagon promoting it and insulin inhibiting it. This ensures that glucose is produced when needed, but not when carbohydrates are abundant. Fatty acid synthesis occurs when the body has an excess of acetyl-CoA, often after consuming a carbohydrate-rich meal. Acetyl-CoA produced in the mitochondria is transported to the cytoplasm, where it serves as the starting material for fatty acid synthesis. The process involves the repeated addition of 2 carbon units to a growing fatty acid chain, catalyzed by a large enzyme complex called fatty acid synthase. Each round of synthesis requires ATP and NADPH. The NADPH is often supplied by the pentose phosphate pathway, an alternative route for glucose oxidation that produces NADPH and ribose 5-phosphate, a precursor for nucleotide synthesis. Fatty acid synthesis continues until a fatty acid of appropriate length is produced, typically 16 carbons. Longer fatty acids can be produced by additional enzymes. These newly synthesized fatty acids can be used to build triglycerides for energy storage or phospholipids for membrane synthesis. Amino acid synthesis is more complex because there are 20 standard amino acids, each with its own biosynthetic pathway. Some amino acids can be synthesized by the human body from common metabolic intermediates. These are called non-essential amino acids, though this term is somewhat misleading. They are essential for life but can be synthesized endogenously. Other amino acids cannot be synthesized by humans and are obtained from the diet. These are the essential amino acids. The carbon skeletons for amino acid synthesis often come from intermediates of glycolysis or the citric acid cycle. For example, pyruvate can be converted to alanine by the addition of an amino group. Alpha-ketoglutarate, an intermediate of the citric acid cycle, can be converted to glutamate. Oxaloacetate can be converted to aspartate. The amino groups are typically supplied by glutamate or glutamine, which act as amino group donors in transamination reactions. These reactions are catalyzed by enzymes called transaminases. The synthesis of nucleotides, the building blocks of DNA and RNA, involves the assembly of a nitrogenous base, a ribose or deoxyribose sugar, and one or more phosphate groups. The pathways for purine and pyrimidine synthesis are distinct but share common themes. Both require amino acids, ribose 5-phosphate and ATP. Both are regulated by feedback inhibition, where the end products of the pathway inhibit the enzymes that catalyze early steps, preventing overproduction. Purine synthesis begins with ribose 5-phosphate, which is converted through a series of 10 steps into inosine monophosphate, a precursor that can be modified to produce adenine and guanine nucleotides. Pyrimidine synthesis begins with the formation of carbomoyl phosphate, which combines with aspartate to form the pyrimidine ring. This ring is then attached to ribose 5-phosphate to form a nucleotide. The pathways are complex, involving many enzymes and intermediates, but the principle is simple. Small molecules are assembled step by step into larger, more complex structures. The pathways of metabolism are interconnected in ways that allow cells to respond flexibly to changing conditions. A molecule like acetyl-CoA sits at a metabolic crossroads. It can be produced from glucose, fatty acids, or amino acids. It can be oxidized in the citric acid cycle to produce energy, or it can be used to synthesize fatty acids, cholesterol, or ketone bodies. The fate of acetyl-CoA depends on the cell's needs and the availability of other nutrients. Similarly, the intermediates of the citric acid cycle are not merely steps in energy production. They are also starting points for biosynthesis. Alpha-ketoglutarate can be used to synthesize glutamate and other amino acids. Succinyl-CoA is used in hemosynthesis, the molecule at the center of hemoglobin. Oxaloacetate can be converted to glucose through gluconeogenesis or to aspartate for protein synthesis. This dual role, both catabolic and anabolic, makes the citric acid cycle a central hub of metabolism, integrating energy production with biosynthesis. The pentose phosphate pathway provides another example of metabolic integration. This pathway branches off from glycolysis using glucose 6-phosphate as its starting point. It has two phases. The oxidative phase generates NADPH and releases carbon dioxide. The non-oxidative phase interconverts various sugar phosphates, eventually producing intermediates that can re-enter glycolysis or be used in nucleotide synthesis. The pentose phosphate pathway is especially active in tissues that require large amounts of NADPH, such as the liver, which synthesizes fatty acids and cholesterol, and red blood cells, which use NADPH to maintain a reduced environment that protects against oxidative damage. The pathway is regulated by the cell's need for NADPH and ribose 5-phosphate. When NADPH is abundant, the oxidative phase slows. When ribose 5-phosphate is needed for nucleotide synthesis, the non-oxidative phase predominates. Regulation of metabolism occurs at multiple levels. Enzyme activity can be controlled by the availability of substrates and cofactors, by the accumulation of products, and by allosteric regulation, where molecules bind to sites other than the active site and change the enzyme's shape and activity. Covalent modification, such as phosphorylation, can rapidly activate or deactivate enzymes. Gene expression can be turned on or off, increasing or decreasing the amount of enzyme present. Hormones coordinate metabolic activity across different tissues, ensuring that the body's needs are met. Feedback inhibition is a common regulatory mechanism. In the pathway for isoleucine synthesis, for example, the end product, isoleucine, inhibits the first enzyme in the pathway. When isoleucine is abundant, the pathway shuts down, preventing wasteful overproduction. When isoleucine is scarce, the inhibition is relieved and synthesis resumes. This type of regulation ensures that resources are used efficiently. Hormonal regulation of metabolism is particularly important in multicellular organisms where different tissues must coordinate their activities. Insulin, released after a meal when blood glucose is high, promotes glucose uptake by cells, stimulates glycolysis and fatty acid synthesis, and inhibits gluconeogenesis and fatty acid oxidation. Glucagon, released when blood glucose is low, has the opposite effects. It stimulates gluconeogenesis and fatty acid oxidation while inhibiting glycolysis and fatty acid synthesis. Epinephrine, also known as adrenaline, is released during stress or exercise. It rapidly mobilizes energy stores, stimulating the breakdown of glycogen and triglycerides. It also increases heart rate and blood flow to muscles, preparing the body for action. These hormonal signals are transmitted through signaling pathways that involve receptors, second messengers, and cascades of phosphorylation events, amplifying the signal and producing a coordinated response. The electron transport chain, which we introduced earlier, deserves a closer look. This chain of protein complexes in the inner mitochondrial membrane is where the majority of ATP is produced. High-energy electrons from NADH and FADH2 enter the chain and are passed from one complex to the next, losing energy at each step. This energy is used to pump protons from the mitochondrial matrix into the intermembrane space, creating a proton gradient. The first complex, NADH dehydrogenase, also called complex 1, accepts electrons from NADH and passes them to ubiquinone, a small lipid-soluble molecule that moves freely within the membrane. As electrons pass through complex 1, 4 protons are pumped across the membrane. Ubiquinone carries the electrons to complex 3, the cytochrome BC1 complex, which pumps 4 more protons. From complex 3, electrons are transferred to cytochrome C, a small protein that moves along the surface of the membrane, carrying electrons to complex 4, cytochrome CE oxidase. Complex 4 transfers the electrons to oxygen, the final electron acceptor, combining them with protons to form water. This step is crucial. Without oxygen to accept the electrons, the entire chain backs up, NADH and FADH2 accumulate, and ATP production stops. This is why oxygen is essential for aerobic life. Complex 4 also pumps 2 protons across the membrane, contributing to the gradient. The proton gradient created by the electron transport chain represents stored energy, a difference in both concentration and charge across the membrane. Protons naturally want to flow back into the matrix, down their concentration gradient. They do so through ATP synthase, a remarkable molecular machine that spans the inner membrane. As protons flow through ATP synthase, the enzyme rotates, and this mechanical rotation drives the synthesis of ATP from ADP and inorganic phosphate. ATP synthase consists of 2 main parts, F0, embedded in the membrane, and F1, protruding into the matrix. The F0 portion forms a channel through which protons flow, causing a rotor to spin. The F1 portion contains the catalytic sites where ATP is synthesized. The rotation of the rotor changes the conformation of the catalytic sites, facilitating the binding of ADP and phosphate, the formation of ATP, and the release of ATP. It is estimated that the synthesis of each ATP requires the flow of about 3 protons through ATP synthase. The efficiency of oxidative phosphorylation depends on the integrity of the inner mitochondrial membrane. If the membrane becomes leaky to protons, the gradient dissipates and ATP synthesis decreases. Some organisms use this to their advantage. Brown adipose tissue found in newborns and hibernating animals contains a protein called thermogenin, or uncoupling protein 1. This protein allows protons to flow back into the matrix without passing through ATP synthase, dissipating the gradient as heat rather than capturing it as ATP. This generates warmth without shivering, helping to maintain body temperature. The citric acid cycle, glycolysis, and the electron transport chain are often presented as distinct pathways, but in reality, they are deeply interconnected and interdependent. Glycolysis produces pyruvate and NADH. Pyruvate is converted to acetyl-CoA, which enters the citric acid cycle. The citric acid cycle produces NADH and FADH2, which feed into the electron transport chain. The electron transport chain produces ATP and regenerates NAD+ and FAD, which are needed for glycolysis and the citric acid cycle to continue. If any part of this system is disrupted, the entire process slows or stops. Consider what happens during intense exercise. Muscles consume ATP rapidly, and the demand for oxygen increases. Breathing rate and heart rate rise to deliver more oxygen to the muscles. Glycolysis speeds up, producing pyruvate faster than the citric acid cycle and electron transport chain can process it. In this situation, pyruvate is converted to lactate, regenerating NAD+ so glycolysis can continue. Lactate accumulates in the muscles and the blood, causing the burning sensation associated with intense effort. When exercise stops and oxygen becomes abundant again, lactate is converted back to pyruvate and oxidized, repaying the oxygen debt. During prolonged fasting or a low-carbohydrate diet, the body adapts by producing ketone bodies, small molecules synthesized from acetyl-CoA in the liver. When glucose is scarce and fatty acid oxidation is high, acetyl-CoA accumulates faster than the citric acid cycle can process it. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA, which is then converted to acetoacetate, a ketone body. Acetoacetate can be reduced to beta-hydroxybutyrate or spontaneously decarboxylated to acetone. Ketone bodies are released into the bloodstream and taken up by tissues like the brain, heart, and skeletal muscle. There, they are converted back to acetyl-CoA and oxidized in the citric acid cycle, providing an alternative fuel when glucose is unavailable. The brain, which normally relies almost exclusively on glucose, can adapt to use ketone bodies for up to 70% of its energy needs during prolonged fasting. This metabolic flexibility is one of the ways the body survives periods of food scarcity. The regulation of ketone body production is tightly linked to hormonal signals. During fasting, insulin levels drop and glucagon levels rise. This stimulates fatty acid oxidation and inhibits fatty acid synthesis, leading to increased acetyl-CoA production. The liver responds by ramping up ketone body synthesis. When food is consumed and insulin levels rise again, fatty acid oxidation slows, acetyl-CoA levels drop, and ketone body production decreases. This hormonal control ensures that ketone bodies are produced only when needed, Glycogen, the storage form of glucose, provides another example of metabolic regulation. Glycogen is a branched polymer of glucose stored primarily in the liver and muscles. When blood glucose levels are high, insulin stimulates glycogen synthesis or glycogenesis. Glucose molecules are added to the growing glycogen chain through the action of glycogen synthase. When blood glucose levels drop, glucagon stimulates glycogen breakdown or glycogenolysis. Glucose molecules are removed from the glycogen chain by the enzyme glycogen phosphorylase, releasing glucose 1-phosphate, which is converted to glucose 6-phosphate. In the liver, glucose 6-phosphate can be dephosphorylated to form free glucose, which is released into the bloodstream to maintain blood glucose levels. In muscles, glucose 6-phosphate cannot be converted to free glucose, so it enters glycolysis and is used locally for energy. This difference reflects the distinct roles of these tissues. The liver helps maintain blood glucose for the benefit of the entire body, while muscles use glycogen to fuel their own activity. The enzymes that control glycogen metabolism, glycogen synthase, and glycogen phosphorylase, are regulated by covalent modification and allosteric effectors. Phosphorylation activates glycogen phosphorylase and inactivates glycogen synthase, promoting glycogen breakdown. Dephosphorylation has the opposite effect, promoting glycogen synthesis. Hormones like insulin, glucagon, and epinephrine trigger cascades of phosphorylation and dephosphorylation events, rapidly switching metabolism between storage and mobilization modes. Cholesterol metabolism provides yet another layer of metabolic complexity. Cholesterol is synthesized from acetyl-CoA through a long series of reactions, primarily in the liver. The pathway produces not only cholesterol but also intermediates used in the synthesis of steroid hormones, bile acids, and vitamin D. The rate-limiting enzyme in cholesterol synthesis is HMG-CoA reductase, which is regulated by cholesterol levels through feedback inhibition and by hormones. When cholesterol is abundant, HMG-CoA reductase is inhibited, slowing cholesterol synthesis. When cholesterol is scarce, the enzyme is activated. Statins, a class of drugs used to lower blood cholesterol, work by inhibiting HMG-CoA reductase, reducing the liver's production of cholesterol and forcing it to take up more cholesterol from the blood. This example illustrates how understanding metabolic pathways can lead to therapeutic interventions. Amino acid metabolism is equally intricate. The body can synthesize about half of the amino acids it needs. The rest are obtained from dietary proteins. When proteins are digested, they are broken down into amino acids, which are absorbed and used to synthesize new proteins, synthesized into other molecules, or catabolized for energy. Amino acids that are not immediately needed are not stored. Instead, excess amino acids are deaminated, and their carbon skeletons are converted to intermediates of the citric acid cycle or to pyruvate, which can be used for energy or converted to glucose or fat. The amino groups removed during deamination are toxic and are converted to urea in the liver through the urea cycle. Urea is then excreted by the kidneys. The urea cycle connects to the citric acid cycle through shared intermediates, illustrating once again the interconnectedness of metabolic pathways. The carbon skeletons of amino acids enter metabolism at different points depending on the amino acid. Some are converted to pyruvate, some to acetyl-CoA, and others to intermediates of the citric acid cycle. Amino acids are classified as glucogenic if their carbon skeletons can be converted to glucose through gluconeogenesis, or ketogenic if they are converted to acetyl-CoA. or acetoacetate and can contribute to ketone body production. Some amino acids are both glucogenic and ketogenic. This classification reflects the metabolic fate of each amino acid and highlights the versatility of amino acid metabolism. We have journeyed through the pathways of energy production and biosynthesis, observing how glucose, fatty acids, and amino acids are broken down and rebuilt, how ATP is generated and consumed, how enzymes catalyze reactions, and how their activity is regulated. We have seen how metabolism is not a rigid sequence of reactions, but a flexible, responsive network that adapts to the needs of the cell and the organism. The pathways interconnect, share intermediates, and are coordinated by hormones and allosteric regulators. As we prepare to continue, let us pause to appreciate the elegance of this molecular choreography. The same principles apply whether we are considering the breakdown of glucose in a muscle cell, the synthesis of fatty acids in the liver, or the production of ketone bodies during fasting. Energy is conserved, not lost. Molecules are transformed, not destroyed. And through it all, life persists, sustained by the ceaseless flow of matter and energy through the pathways of metabolism. In the next part of our journey, we will explore how cells communicate with each other, how signals are transmitted, and how genetic information is expressed and regulated, building toward a more complete understanding of the integrated system that is biochemistry. We have traveled through the molecular landscape of metabolism, observing how energy flows through the breakdown and synthesis of carbohydrates, lipids, and proteins. We have seen how pathways interconnect, how enzymes guide reactions with precision, and how cells adapt their chemistry to meet changing needs. Now, we shift our focus toward the ways cells communicate, both with their environment and with each other, and how genetic information is stored, expressed, and regulated. These processes reveal another dimension of biochemistry, one where molecules carry messages, where information flows as surely as energy does, and where the dance of chemistry gives rise to the complexity of life. Communication at the molecular level begins with signals. A signal may be a hormone released into the bloodstream, a neurotransmitter crossing the gap between nerve cells, or a molecule on the surface of one cell binding to a receptor on another. Regardless of the form, the principle is the same. A signaling molecule carries information from one location to another, and when it reaches its target, it triggers a response. This response may involve changes in metabolism, gene expression, cell movement, or cell division. Through signaling, cells coordinate their activities, allowing tissues and organs to function as integrated systems. Cell signaling typically involves 3 stages. Reception, where the signal binds to a receptor. Transduction, where the signal is converted into a form that can bring about a cellular response. And response, where the cell carries out the action specified by the signal. Each stage involves specific molecules and mechanisms, and together they form pathways that amplify and refine the original signal. Receptors are proteins that bind specifically to signaling molecules, often called ligands. The binding of a ligand to a receptor changes the receptor's shape, triggering a cascade of events inside the cell. Receptors can be located on the cell surface or inside the cell, depending on the nature of the signal. Hydrophilic signals, like most hormones and neurotransmitters, cannot cross the lipid bilayer of the cell membrane, so their receptors are embedded in the membrane. Hydrophobic signals, like steroid hormones, can pass through the membrane and bind to receptors inside the cell. G-protein coupled receptors are a large family of cell surface receptors involved in many signaling pathways. When a ligand binds to a G-protein coupled receptor, the receptor activates a G-protein, a molecular switch that cycles between an inactive state bound to GDP and an active state bound to GTP. The activated G-protein then interacts with other proteins, often enzymes that produce second messengers, small molecules that relay the signal inside the cell. One common second messenger is cyclic AMP or CAMP, produced from ATP by the enzyme adenylyl cyclase. When a G protein activates adenylyl cyclase, CAMP levels inside the cell rise. CAMP binds to and activates an enzyme called protein kinase A, which phosphorylates target proteins, changing their activity. The targets of protein kinase A vary depending on the cell type, but they often include enzymes involved in metabolism. In this way, a signal received at the cell surface can rapidly alter metabolic activity inside the cell. Another second messenger is calcium. Calcium ions are kept at very low concentrations in the cytoplasm, much lower than in the extracellular fluid or in certain organelles like the endoplasmic reticulum. When a signal opens calcium channels, calcium floods into the cytoplasm, and this sudden increase in concentration serves as a signal. Calcium binds to proteins like calmodulin, which then activate other proteins, including kinases and phosphatases. Calcium signaling is involved in processes ranging from muscle contraction to the release of neurotransmitters to the activation of immune cells. Receptor tyrosine kinases are another major class of cell surface receptors. When a ligand binds to a receptor tyrosine kinase, the receptor dimerizes, meaning two receptor molecules come together. This brings their intracellular domains close enough to phosphorylate each other on tyrosine residues. These phosphorylated tyrosines serve as docking sites for other proteins, initiating signaling cascades. Receptor tyrosine kinases are involved in regulating cell growth, differentiation, and survival. Growth factors like insulin and epidermal growth factor signal through receptor tyrosine kinases. One important signaling pathway activated by receptor tyrosine kinases is the MAP kinase pathway. MAP stands for mitogen activated protein. This pathway involves a cascade of kinases, each one phosphorylating and activating the next. The final kinase in the chain, MAP kinase, enters the nucleus and phosphorylates transcription factors, proteins that regulate gene expression. In this way, a signal received at the cell surface can lead to changes in which genes are turned on or off, altering the cell's behavior over the long term. Signaling pathways often involve amplification. A single ligand molecule binding to a receptor can activate many G proteins. Each G protein can activate many adenylyl cyclase molecules. Each adenylyl cyclase can produce many CAMP molecules. Each CAMP can activate a protein kinase A, and each kinase can phosphorylate many target proteins. This cascade amplifies the original signal, allowing a small stimulus to produce a large response. Amplification is one reason why hormones can be effective at very low concentrations. Signaling pathways also interact with each other, a phenomenon called crosstalk. A signal that activates one pathway may enhance or inhibit another. This allows cells to integrate multiple signals and produce appropriate responses. For example, insulin signaling and growth factor signaling share some of the same downstream components, allowing the cell to coordinate growth with nutrient availability. Crosstalk adds another layer of complexity to cell signaling, making it a flexible and responsive system. Termination of the signal is as important as its initiation. Signals cannot be allowed to persist indefinitely, or the cell would lose the ability to respond to new information. Second messengers are rapidly degraded or removed. CAMP is broken down by an enzyme called phosphodiesterase. Calcium is pumped back out of the cytoplasm. Phosphate groups added by kinases are removed by phosphatases, reversing the signal. Receptors can be internalized, removing them from the cell surface. These mechanisms ensure that signaling is a dynamic process, constantly turning on and off in response to changing conditions. Steroid hormones, being lipid-soluble, take a different route. They diffuse through the cell membrane and bind to receptors in the cytoplasm or nucleus. The hormone receptor complex then binds to specific DNA sequences, acting as a transcription factor to regulate gene expression. This direct regulation of transcription allows steroid hormones to produce long-lasting changes in cell behavior. Examples include estrogen, testosterone, and cortisol, each of which regulates a wide range of physiological processes. The ability of cells to respond to signals depends on the presence of the appropriate receptors. Different cell types express different sets of receptors, allowing them to respond selectively to signals in their environment. A hormone circulating in the bloodstream reaches many tissues, but only those with the corresponding receptor will respond. This specificity is crucial for coordinating the activities of different tissues. Insulin, for example, affects muscle, liver, and adipose tissue, but not all tissues in the same way. Each tissue expresses the insulin receptor and responds according to its own metabolic needs. Genetic information stored in DNA is the blueprint for building and maintaining an organism. The sequence of nucleotides in DNA encodes the sequence of amino acids in proteins, and proteins carry out most of the work of the cell. The flow of information from DNA to RNA to protein is central to biology. This process, sometimes called the central dogma, involves two main steps: transcription, where DNA is copied into RNA, and translation, where RNA is used to synthesize proteins. Transcription occurs in the nucleus. The enzyme RNA polymerase binds to a region of DNA called a promoter located near the beginning of a gene. The DNA double helix unwinds and RNA polymerase moves along the template strand, synthesizing a complementary strand of RNA. The RNA nucleotides pair with the DNA nucleotides according to base pairing rules. Adenine pairs with uracil in RNA, and guanine pairs with cytosine. The result is a molecule of messenger RNA, or mRNA, that carries a copy of the gene's information. In eukaryotic cells, the initial RNA transcript, called pre-mRNA, undergoes processing before it can be translated. A cap is added to the 5' end, and a poly-A tail is added to the 3' end. These modifications protect the mRNA from degradation and assist in translation. The pre-mRNA also contains introns, non-coding sequences that are removed by a process called splicing. The remaining sequences, called exons, are joined together to form the mature mRNA. Splicing allows a single gene to produce multiple different proteins through alternative splicing, where different combinations of exons are included in the final mRNA. Once processing is complete, the mRNA is exported from the nucleus to the cytoplasm, where translation occurs. Translation is carried out by ribosomes, complex molecular machines composed of ribosomal RNA and proteins. The ribosome reads the mRNA sequence in units of 3 nucleotides called codons. Each codon specifies a particular amino acid or a stop signal. Transfer RNA or tRNA molecules act as adapters, each one carrying a specific amino acid and recognizing a specific codon through a complementary sequence called an anticodon. Translation begins when a ribosome binds to the mRNA near the start codon, typically AUG, which codes for methionine. A tRNA carrying methionine binds to the start codon, and the ribosome begins moving along the mRNA, reading each codon in sequence. For each codon, the appropriate tRNA binds, delivering its amino acid. The ribosome catalyzes the formation of a peptide bond between the growing polypeptide chain and the new amino acid. The tRNA is then released, and the ribosome moves to the next codon. This continues until a stop codon is reached, at which point the ribosome releases the completed polypeptide. The genetic code, the correspondence between codons and amino acids, is nearly universal, shared by almost all living organisms. There are 64 possible codons, but only 20 standard amino acids. This means the code is redundant. Most amino acids are specified by more than one codon. For example, leucine is specified by 6 different codons. This redundancy provides some protection against mutations. A change in the third position of a codon often does not change the amino acid, so the protein remains functional. Mutations, changes in the DNA sequence, can have various effects. Some mutations are silent, causing no change in the protein. Some are missense mutations, changing one amino acid to another, which may or may not affect protein function. Some are nonsense mutations, changing an amino acid codon to a stop codon, resulting in a truncated protein. And some are frameshift mutations, caused by insertions or deletions that shift the reading frame, often producing a completely non-functional protein. Mutations are the raw material of evolution, introducing variation on which natural selection can act. Gene expression, the process by which information in a gene is used to synthesize a functional product, is regulated at multiple levels. Not all genes are expressed in all cells or at all times. Regulation allows cells to respond to their environment, differentiate into specialized types, and coordinate their activities. Much of this regulation occurs at the level of transcription, determining whether a gene is transcribed into RNA. Transcription factors are proteins that bind to specific DNA sequences near genes and either promote or inhibit transcription. Some transcription factors are activators, enhancing the binding of RNA polymerase and increasing transcription. Others are repressors, blocking RNA polymerase or recruiting proteins that condense the DNA, making it inaccessible. The combination of transcription factors present in a cell determines which genes are active. Eukaryotic DNA is packaged with proteins called histones, forming a structure called chromatin. The degree of chromatin condensation affects gene expression. Tightly packed chromatin is generally inactive, while loosely packed chromatin is accessible to transcription machinery. Chemical modifications to histones, such as acetylation and methylation, can alter chromatin structure and regulate gene expression. Acetylation typically loosens chromatin, promoting transcription, while methylation can either activate or repress transcription, depending on the context. DNA methylation, the addition of methyl groups to cytosine bases, is another mechanism of gene regulation. In many organisms, including mammals, methylation of promoter regions is associated with gene silencing. Methylation patterns can be inherited through cell divisions, allowing cells to maintain their identity even as they divide. This form of regulation, which does not change the DNA sequence, is called epigenetics. Epigenetic modifications play important roles in development, allowing cells with identical DNA to differentiate into diverse cell types. Post-transcriptional regulation provides additional control. The stability of mRNA affects how much protein is produced. Some mRNAs are rapidly degraded, while others persist for hours or days. The rate of translation can also be regulated. Small RNA molecules such as microRNAs can bind to mRNAs and either block translation or target the mRNA for degradation. This adds another layer of control, allowing cells to fine-tune protein levels without changing transcription rates. Proteins themselves can be regulated after synthesis. Many proteins are synthesized as inactive precursors and are activated by cleavage or by the addition of chemical groups. Phosphorylation, as we have seen, is a common mechanism for regulating enzyme activity. Other modifications such as acetylation, methylation, and ubiquitination also play regulatory roles. Ubiquitination, the addition of a small protein called ubiquitin, often marks proteins for degradation by the proteasome, a large protein complex that breaks down proteins. This allows cells to rapidly remove proteins that are no longer needed or that are damaged. The regulation of gene expression is not static. It responds dynamically to signals from the environment and from other cells. When a cell receives a signal, such as a hormone or growth factor, the signal is transduced through a pathway that often culminates in the activation or inhibition of transcription factors. These transcription factors then alter the expression of target genes, producing a response. In this way, signaling and gene regulation are intimately connected. Consider the response to low oxygen, a condition called hypoxia. When oxygen levels drop, a transcription factor called hypoxia-inducible factor, or HIF, is stabilized and activates genes involved in oxygen delivery and metabolic adaptation. These genes include those encoding enzymes for glycolysis, which does not require oxygen, and vascular endothelial growth factor, which promotes the formation of new blood vessels. When oxygen levels return to normal, HIF is degraded and the response is turned off. This illustrates how gene regulation allows cells to adapt to changing conditions. The lactoperon in bacteria provides a classic example of gene regulation, though it differs from eukaryotic mechanisms. The lactoperon consists of genes encoding enzymes for lactose metabolism, along with regulatory sequences. When lactose is absent, a repressor protein binds to the operator sequence, blocking transcription. When lactose is present, it binds to the repressor, causing it to release from the DNA, allowing transcription to proceed. This simple on-off switch ensures that the enzymes are produced only when lactose is available. Eukaryotic gene regulation is more complex, involving multiple transcription factors, chromatin modifications, and post-transcriptional mechanisms. Enhancers, DNA sequences that can be located far from the genes they regulate, bind transcription factors and increase transcription. Silencers have the opposite effect. The three-dimensional structure of chromatin brings enhancers and promoters into proximity, allowing regulatory proteins to interact. This spatial organization adds another dimension to gene regulation. Development provides a compelling illustration of gene regulation. A fertilized egg contains all the genetic information needed to build an entire organism. Yet different cells express different sets of genes. As the embryo develops, cells receive signals that activate specific transcription factors, which in turn activate other genes, creating cascades of gene expression that define cell fate. Gradients of signaling molecules provide positional information, telling cells where they are in the developing embryo and what type of cell they will become. Homeotic genes, also called Hox genes, are master regulators of development in animals. They encode transcription factors that control the expression of many other genes, determining the identity of body segments. Mutations in Hox genes can cause dramatic changes, such as the development of legs where antennae should be in fruit flies. The conservation of Hox genes across species underscores their fundamental importance. Similar genes are found in organisms as diverse as insects, fish, and humans, suggesting they arose early in animal evolution. Cells also regulate their response to DNA damage. DNA is constantly subjected to damaging agents, including ultraviolet light, reactive oxygen species, and errors during replication. If left unrepaired, DNA damage can lead to mutations, cell dysfunction, or cancer. Cells have evolved sophisticated mechanisms for detecting and repairing DNA damage. When damage is detected, checkpoint proteins halt the cell cycle, giving the cell time to repair the damage before proceeding. If the damage is too severe, the cell may activate programmed cell death or apoptosis to prevent the propagation of damaged DNA. Apoptosis is a carefully regulated process of cell suicide. It involves a cascade of proteases called caspases that systematically dismantle the cell. The cell shrinks, the DNA is fragmented, and the cell is packaged into small vesicles that are consumed by neighboring cells. Apoptosis occurs naturally during development, sculpting tissues, and removing cells that are no longer needed. It also serves as a defense against cancer, eliminating cells that have acquired dangerous mutations. The decision to undergo apoptosis is influenced by signals from both inside and outside the cell. Cells receive survival signals from neighboring cells, and the absence of these signals can trigger apoptosis. Internal damage, such as irreparable DNA damage or severe stress, can also activate apoptosis. The balance between pro-apoptotic and anti-apoptotic proteins determines whether a cell lives or dies. Cancer cells often evade apoptosis, acquiring mutations that tip the balance in favor of survival, allowing them to proliferate unchecked. The cell cycle, the sequence of events by which a cell grows and divides, is also tightly regulated. The cycle consists of phases. G1, where the cell grows and prepares for DNA replication. S phase, where DNA is replicated. G2, where the cell prepares for division. And M phase, where the cell divides into two daughter cells. Checkpoints ensure that each phase is completed correctly before the next begins. Cyclins and cyclin-dependent kinases, or CDKs, are key regulators of the cell cycle. Cyclins are proteins whose levels rise and fall during the cycle. CDKs are kinases that are active only when bound to a cyclin. Different cyclin-CDK complexes drive different phases of the cycle. The levels of cyclins are regulated by synthesis and degradation, ensuring that each phase occurs in the proper order. Mutations that disrupt cell cycle regulation can lead to uncontrolled cell division, a hallmark of cancer. Cancer is fundamentally a disease of gene regulation and cell signaling. Cancer cells acquire mutations that allow them to divide uncontrollably, evade apoptosis, and invade other tissues. Oncogenes are mutated versions of normal genes that promote cell division. Tumor suppressor genes, when functioning normally, inhibit cell division or promote apoptosis. Mutations that inactivate tumor suppressor genes remove these breaks, allowing cells to proliferate. Most cancers require multiple mutations affecting both oncogenes and tumor suppressor genes, as well as genes involved in DNA repair and other processes. Understanding the biochemical basis of cancer has led to targeted therapies. Some cancer drugs inhibit specific enzymes or receptors that are overactive in cancer cells. Others target signaling pathways that cancer cells rely on. Immunotherapies enhance the immune system's ability to recognize and destroy cancer cells. These approaches illustrate how detailed knowledge of biochemistry can translate into medical applications. Metabolism, signaling, and gene regulation are not separate domains. They are intertwined, each influencing the others. Metabolic state affects signaling pathways. For example, the energy sensor AMPK is activated when ATP levels are low. AMPK then activates pathways that produce ATP, such as fatty acid oxidation, and inhibits pathways that consume ATP, such as fatty acid synthesis. AMPK also affects gene expression, promoting the transcription of genes involved in energy production. Conversely, signaling affects metabolism. Insulin signaling, as we have seen, promotes glucose uptake and storage while inhibiting glucose production. Growth factor signaling promotes the synthesis of proteins and lipids, supporting cell growth. Nutrient availability affects gene expression. When amino acids are abundant, a signaling pathway called mTOR is activated, promoting protein synthesis and cell growth. When nutrients are scarce, mTOR is inhibited, and cells activate autophagy, a process of self-digestion where cellular components are broken down and recycled. Autophagy is a regulated process where portions of the cytoplasm, including damaged organelles and aggregated proteins, are enclosed in double membrane vesicles called autophagosomes. These fuse with lysosomes, organelles containing digestive enzymes, and the contents are degraded. The resulting small molecules are released back into the cytoplasm and can be used for energy or biosynthesis. Autophagy is a survival mechanism during starvation, but it also plays roles in quality control, removing damaged components and maintaining cellular health. The circadian rhythm, the internal clock that regulates daily cycles of activity and rest, is another example of integrated regulation. Circadian rhythms are driven by oscillations in gene expression, controlled by a small number of clock genes. These genes encode transcription factors that regulate each other in feedback loops. The protein products of clock genes accumulate during the day, and at night they inhibit their own transcription. This creates a cycle with a period of approximately 24 hours. Circadian rhythms influence metabolism, with enzymes involved in energy production and storage showing daily oscillations in expression and activity. The timing of meals can affect metabolic health, and disruptions to circadian rhythms, such as shift work or jet lag, are associated with metabolic disorders. This highlights how deeply biochemical processes are embedded in time, responding not only to immediate conditions but also to predictable daily cycles. The microbiome, the community of microorganisms living in and on our bodies, also interacts with our biochemistry. Bacteria in the gut help digest complex carbohydrates that our own enzymes cannot break down, producing short-chain fatty acids that we absorb and use for energy. They synthesize vitamins, such as vitamin K and certain B vitamins, that we cannot make ourselves. They train the immune system, helping it distinguish between harmful pathogens and harmless or beneficial microbes. The microbiome's influence extends to metabolism, immune function, and even behavior, illustrating how biochemistry is not confined to a single organism but involves interactions across species. We have explored the flow of information in cells, from signals received at the surface to changes in gene expression and protein activity. We have seen how transcription and translation convert genetic information into functional proteins, and how gene expression is regulated at multiple levels to allow cells to respond to their environment and coordinate their activities. We have observed how signaling pathways and metabolic pathways are interconnected, each influencing the other, creating a dynamic, integrated system. As we continue, we will bring these threads together, examining how all of these processes, from the smallest chemical reaction to the largest regulatory network, unite to form the living, breathing, adapting entity that is a cell, and how cells in turn unite to form tissues, organs, and organisms. The final part of our journey will draw these connections, revealing the unity that underlies the diversity of biochemistry. We have journeyed far through the molecular world, observing the building blocks of life, the pathways of metabolism, the signals that coordinate cellular activity, and the expression of genetic information. We have seen enzymes catalyze reactions with precision, membranes separate inside from outside, energy flow through ATP, and information flow from DNA to RNA to protein. Now, as we approach the completion of our exploration, we turn toward synthesis, bringing these elements together to see how they form an integrated whole, how biochemistry extends beyond individual molecules and pathways to create the phenomenon we call life. At its heart, biochemistry reveals a profound unity. Despite the enormous diversity of living organisms, from bacteria to plants to animals, the fundamental chemistry is remarkably similar. The same nucleotides build DNA in all organisms. The same amino acids construct proteins. The same metabolic pathways, glycolysis, and the citric acid cycle operate in nearly all cells. The genetic code is nearly universal. This unity suggests a common origin, that all life on Earth descended from a single ancestor that possessed these biochemical systems billions of years ago. The origin of life remains one of biology's deepest questions, but biochemistry provides clues. Life requires three fundamental capabilities: the ability to store and transmit information, the ability to catalyze chemical reactions, and the ability to maintain a boundary separating inside from outside. Modern cells use DNA for information storage, proteins for catalysis, and lipid membranes for boundaries. But this creates a puzzle. DNA requires proteins to replicate, and proteins require DNA to specify their sequences. Which came first? The discovery that RNA can both store information and catalyze reactions suggests a possible answer. In the early stages of life, RNA may have served both roles, storing genetic information and catalyzing its own replication. Over time, DNA, being more stable, took over information storage, and proteins, being more versatile, took over catalysis. This RNA world hypothesis is supported by the observation that ribosomes, the machines that synthesize proteins, are made largely of RNA, and that many cofactors, such as ATP and NAD+, contain nucleotide components, suggesting ancient RNA origins. The first cells were likely simple, with a minimal set of biochemical pathways. Over billions of years, these pathways were refined, duplicated, and modified, creating the complexity we see today. Evolution operates on biochemistry just as it does on anatomy or behavior. Mutations introduce variation in proteins and regulatory sequences. Natural selection favors variants that enhance survival and reproduction. Over time, this process shapes the biochemical machinery of life, optimizing it for diverse environments and lifestyles. The universality of biochemistry is not absolute. Evolution has introduced variations on common themes. Some organisms have modified the genetic code, using certain codons to specify different amino acids or using additional amino acids beyond the standard 20. Some have evolved alternative metabolic pathways to exploit unusual energy sources, such as hydrogen gas, iron, or sulfur compounds. Extremophiles, organisms that thrive in harsh environments like hot springs, deep sea vents, or highly acidic waters, have adapted their biochemistry to function under conditions that would denature most proteins and disrupt most membranes. Thermophilic bacteria, for example, have proteins that remain stable at temperatures exceeding the boiling point of water. Their membranes contain lipids with structures that prevent melting at high temperatures. Psychrophiles, organisms adapted to cold environments, have proteins that remain flexible at near freezing temperatures and produce antifreeze compounds to prevent ice crystal formation. Halophiles live in extremely salty environments and maintain osmotic balance by accumulating high concentrations of compatible solutes. These adaptations illustrate the flexibility of biochemistry, its ability to be tuned and modified while retaining core functions. The compartmentalization of biochemistry in eukaryotic cells adds another layer of organization. Eukaryotic cells contain membrane-bound organelles, each specialized for particular functions. The nucleus houses DNA and is the site of transcription. The endoplasmic reticulum is involved in protein and lipid synthesis. The Golgi apparatus processes and packages proteins for transport. Mitochondria produce ATP through oxidative phosphorylation. Chloroplasts in plant cells carry out photosynthesis. Lysosomes contain digestive enzymes for breaking down macromolecules. Peroxisomes break down fatty acids and detoxify harmful substances. This compartmentalization allows incompatible processes to occur simultaneously in different locations. For example, fatty acid synthesis occurs in the cytoplasm, while fatty acid oxidation occurs in mitochondria. By separating these opposing pathways, the cell can regulate them independently, preventing futile cycles where molecules are simultaneously synthesized and degraded. Compartmentalization also creates specialized environments. The interior of mitochondria is more acidic than the cytoplasm, and this pH difference is essential for ATP synthesis. The interior of lysosomes is even more acidic, providing the optimal pH for digestive enzymes. The endoplasmic reticulum, or ER, is a network of membranous tubules and sacs extending throughout the cytoplasm. The rough ER, studded with ribosomes, is where proteins destined for secretion or for insertion into membranes are synthesized. As a protein is synthesized, it is threaded into the ER, where it folds and may be modified by the addition of carbohydrate chains, a process called glycosylation. The smooth ER, lacking ribosomes, is involved in lipid synthesis and in the metabolism of drugs and toxins. Proteins synthesized in the rough ER are transported to the Golgi apparatus, a stack of flattened membranous sacs. The Golgi acts as a sorting and processing center. Proteins move through the Golgi from one side to the other, and at each stage they may be further modified. Carbohydrate chains may be trimmed or extended. Proteins may be cleaved to activate them. Eventually, proteins are packaged into vesicles and sent to their final destinations. whether the cell membrane, secretion to the outside, or delivery to lysosomes. This system of membranes and vesicles allows eukaryotic cells to produce and secrete large amounts of protein. Cells that specialize in secretion, such as pancreatic cells that produce digestive enzymes or plasma cells that produce antibodies, have highly developed ER and Golgi systems. The coordination of protein synthesis, folding, modification, and transport is a remarkable feat of biochemical organization. Mitochondria and chloroplasts are thought to have originated from free-living bacteria that were engulfed by ancestral eukaryotic cells in a process called endosymbiosis. Evidence for this includes the fact that these organelles have their own DNA, separate from the nuclear DNA, and their own ribosomes, which resemble bacterial ribosomes more than eukaryotic ribosomes. The DNA in mitochondria and chloroplasts encodes some, but not all, of the proteins these organelles need. Most mitochondrial and chloroplast proteins are encoded by nuclear genes, synthesized in the cytoplasm and imported into the organelles. This division of labor between nuclear and organellar genes reflects a long evolutionary history of gene transfer from the organelles to the nucleus. The retention of organellar genomes may be necessary because some proteins are too hydrophobic to be imported and are synthesized locally. The coordination between nuclear and organellar gene expression is essential for organelle function and represents another level of regulation in eukaryotic cells. The cytoskeleton, a network of protein filaments extending throughout the cytoplasm, provides structural support and enables movement. Three main types of filaments make up the cytoskeleton. Microtubules, hollow tubes made of the protein tubulin, serve as tracks for motor proteins that transport vesicles and organelles. They also form the mitotic spindle that separates chromosomes during cell division. Actin filaments, made of the protein actin, are involved in cell movement, shape changes, and muscle contraction. Intermediate filaments provide mechanical strength and anchor organelles in place. Motor proteins, such as kinesin and dynein, move along microtubules, carrying cargo toward the plus or minus end of the filament. Myosin motors move along actin filaments and are responsible for muscle contraction. These motor proteins convert the chemical energy of ATP into mechanical work, demonstrating how biochemistry gives rise to physical movement. The coordinated activity of cytoskeletal filaments and motor proteins underlies processes as diverse as cell division, intracellular transport, and the crawling of white blood cells toward sites of infection. The extracellular matrix, the network of proteins and polysaccharides surrounding cells and tissues, also plays important biochemical roles. In animals, the extracellular matrix is composed largely of collagen, a fibrous protein that provides tensile strength, and proteoglycans, proteins attached to long polysaccharide chains that provide compressive resistance. The matrix is not merely a passive scaffold. It binds growth factors and other signaling molecules, modulating their availability to cells. Cells interact with the matrix through integrins, transmembrane receptors that connect the cytoskeleton to the extracellular matrix, allowing cells to sense and respond to their mechanical environment. In plants, the cell wall serves a similar role, providing structural support and protection. Plant cell walls are composed primarily of cellulose, a polysaccharide made of glucose, along with other polysaccharides and proteins. The rigidity of the cell wall allows plants to stand upright without a skeleton. It also prevents cells from bursting when water enters by osmosis, allowing plants to maintain turgor pressure, which is essential for growth and structural integrity. Biochemical communication extends beyond individual cells to coordination at the level of tissues and organs. In multicellular organisms, cells must not only respond to their own internal state, but also to signals from distant cells. Endocrine signaling involves hormones released into the bloodstream, traveling throughout the body to reach target cells. Paracrine signaling involves signals that diffuse to nearby cells. Autocrine signaling involves cells responding to signals they themselves produce. And synaptic signaling, used by nerve cells, involves neurotransmitters released at synapses, the junctions between neurons. The nervous system provides a striking example of specialized biochemical communication. Neurons transmit signals as electrical impulses, changes in membrane potential that propagate along the axon, the long extension of the neuron. When the impulse reaches the synapse, it triggers the release of neurotransmitters, small molecules stored in vesicles. Neurotransmitters diffuse across the synaptic cleft and bind to receptors on the next neuron, generating a new electrical signal. Common neurotransmitters include acetylcholine, dopamine, serotonin, and glutamate. The rapid synthesis, release, and degradation of neurotransmitters allow neurons to transmit information quickly and precisely. Enzymes in the synaptic cleft break down neurotransmitters shortly after release, ensuring that signals are brief and can be repeated rapidly. Reuptake transporters in the presynaptic neuron take neurotransmitters back into the cell, where they can be repackaged into vesicles or degraded. Disruptions in neurotransmitter systems are implicated in many neurological and psychiatric disorders, highlighting the importance of biochemical balance in brain function. The immune system provides another example of biochemical coordination at the organismal level. Immune cells recognize pathogens through receptors that bind to molecules found on bacteria, viruses, and other invaders. When a pathogen is detected, immune cells release cytokines, signaling molecules that recruit other immune cells and activate inflammatory responses. Antibodies, proteins produced by B cells, bind to pathogens and mark them for destruction. The complement system, a cascade of proteins in the blood, forms pores in bacterial membranes, leading to their lysis. The adaptive immune system has the remarkable ability to remember past infections. After an infection is cleared, some immune cells remain as memory cells. If the same pathogen is encountered again, these memory cells respond more quickly and effectively, providing immunity. This immunological memory is the basis for vaccination, where exposure to a harmless form of a pathogen trains the immune system to recognize and respond to the real pathogen. Biochemistry also underlies sensory perception. Vision begins when light strikes the retina, where photoreceptor cells contain the protein rhodopsin. Rhodopsin consists of a protein called opsin bound to retinal, a molecule derived from vitamin A. When retinal absorbs light, it changes shape, triggering a cascade of biochemical events that ultimately produce an electrical signal transmitted to the brain. This signal transduction pathway involves G proteins and cyclic nucleotides, similar in principle to other signaling pathways we have discussed. Taste and smell involve receptors that bind to specific molecules. Taste receptors on the tongue detect sweet, sour, salty, bitter, and umami flavors. Olfactory receptors in the nose detect a vast array of odor molecules. Binding of a molecule to its receptor initiates a signaling cascade that generates a nerve impulse. The brain integrates signals from many receptors to create the perception of taste or smell. the biochemistry of sensation transforms chemical information into neural information, which the brain interprets as sensory experience. Hormonal regulation integrates metabolism across the body. We have discussed insulin and glucagon, which regulate blood glucose levels. Other hormones coordinate growth, reproduction, stress responses, and circadian rhythms. Growth hormone stimulates protein synthesis and cell division, promoting growth during childhood and adolescence. Thyroid hormones regulate metabolic rate, affecting how quickly cells produce and consume energy. Cortisol, released during stress, mobilizes energy stores and suppresses non-essential processes like digestion and immune function. The hypothalamus and pituitary gland, located in the brain, serve as master regulators of the endocrine system. The hypothalamus produces hormones that control the pituitary, which in turn releases hormones that regulate other endocrine glands throughout the body. This hierarchical organization allows the nervous system to influence the endocrine system, integrating neural and hormonal control. The hypothalamic pituitary adrenal axis, for example, coordinates the stress response, linking perception of a threat to the release of cortisol. Aging is another process with biochemical roots. Over time, cells accumulate damage. DNA acquires mutations. Proteins become oxidized or glycated, forming crosslinks that impair function. Mitochondria become less efficient, producing more reactive oxygen species and less ATP. Telomeres, protective sequences at the ends of chromosomes, shorten with each cell division, eventually limiting the cell's ability to divide. The accumulation of senescent cells, cells that have stopped dividing but remain metabolically active, contributes to inflammation and tissue dysfunction. Understanding the biochemical basis of aging has led to research into interventions that might slow the aging process. Caloric restriction, reducing calorie intake without malnutrition, extends lifespan in many organisms and is associated with improved metabolic health. The mechanisms involve activation of sirtuins, proteins that promote DNA repair and mitochondrial function, and inhibition of mTOR, reducing protein synthesis and promoting autophagy. Whether these interventions translate to humans remains an active area of research, but they illustrate how biochemical knowledge can inform approaches to health and longevity. Exercise provides another example of how biochemistry influences health. Regular physical activity increases mitochondrial density in muscle cells, enhancing their capacity for aerobic metabolism. It improves insulin sensitivity, allowing cells to take up glucose more effectively. It stimulates the release of factors that promote brain health, including brain-derived neurotrophic factor, which supports neuronal growth and survival. These biochemical adaptations contribute to the wide-ranging health benefits of exercise. Nutrition is fundamentally a matter of biochemistry. The nutrients we consume provide the raw materials and energy needed for metabolism. Carbohydrates provide glucose for energy. Fats provide concentrated energy and essential fatty acids. Proteins provide amino acids for protein synthesis. Vitamins and minerals serve as cofactors for enzymes and structural components of molecules. Deficiencies in any of these nutrients disrupt metabolic pathways and can lead to disease. Vitamin deficiencies illustrate the importance of cofactors. Vitamin C is required for the hydroxylation of proline and lysine in collagen synthesis. Without it, collagen is unstable, leading to scurvy, characterized by bleeding gums and poor wound healing. Vitamin B1, thiamine, is required for enzymes in carbohydrate metabolism. Deficiency causes beriberi, with symptoms including muscle weakness and nerve damage. Vitamin D is required for calcium absorption. Deficiency leads to rickets in children and osteomalacia in adults, both characterized by weak bones. The interaction between diet and metabolism extends to the microbiome. The bacteria in our gut metabolize dietary fiber, producing short-chain fatty acids like butyrate, which serves as fuel for intestinal cells and has anti-inflammatory effects. They also metabolize bile acids, influencing cholesterol metabolism. Dietary composition affects the composition of the microbiome, and changes in the microbiome can affect metabolism, immunity, and even behavior. This bidirectional relationship highlights how biochemistry extends beyond the individual organism to include interactions with microbial communities. Pharmacology, the study of drugs, is applied biochemistry. Most drugs work by interacting with specific proteins, such as enzymes, receptors, or ion channels. Understanding the biochemical basis of disease allows for the design of drugs that target specific molecular defects. Enzyme inhibitors, for example, can block overactive enzymes. Receptor agonists mimic natural ligands, activating receptors, while receptor antagonists block receptors, preventing activation. Aspirin, one of the oldest and most widely used drugs, works by inhibiting an enzyme called cyclooxygenase, which is involved in the synthesis of prostaglandins, signaling molecules that promote inflammation, pain, and fever. By blocking this enzyme, aspirin reduces these symptoms. Statins, used to lower cholesterol, inhibit HMG-CoA reductase, the rate-limiting enzyme in cholesterol synthesis. Antibiotics target bacterial enzymes or structures that are not present in human cells, allowing them to kill bacteria without harming the host. The development of new drugs increasingly relies on detailed knowledge of protein structure and function. Rational drug design uses information about the three-dimensional shape of a target protein to design molecules that bind tightly and specifically. Computational methods can predict how candidate drugs will interact with their targets, accelerating the discovery process. Advances in genomics allow for personalized medicine, where treatment is tailored to an individual's genetic makeup, predicting which drugs will be most effective and which side effects are most likely. Biotechnology harnesses biochemistry for practical applications. Recombinant DNA technology allows genes to be transferred between organisms, enabling the production of human proteins in bacteria or yeast. Insulin, once extracted from animal pancreases, is now produced by genetically engineered bacteria, making it more accessible and free of animal contaminants. Other therapeutic proteins, such as clotting factors for hemophilia and growth hormone for growth disorders, are produced similarly. Gene editing technologies, such as CRISPR-Cas9, allow precise modifications to DNA sequences. This has applications in research, agriculture, and medicine. Researchers use gene editing to study gene function by creating knockout organisms with specific genes disabled. In agriculture, gene editing can create crops with improved yield, nutritional content, or resistance to pests and disease. In medicine, gene editing holds promise for correcting genetic defects, potentially curing inherited diseases. Synthetic biology takes biotechnology further, designing and constructing new biological systems from scratch. Researchers have created minimal genomes, stripping away non-essential genes to create the simplest possible living cell. They have engineered bacteria to produce biofuels, pharmaceuticals, and materials. They have designed genetic circuits that function like electronic circuits, responding to inputs with predictable outputs. These advances blur the line between biology and engineering, raising both exciting possibilities and ethical questions. The biochemical perspective on life emphasizes processes over structures, flows over states. A cell is not a static collection of molecules, but a dynamic system where matter and energy flow continuously. Molecules are constantly being synthesized and degraded, modified and transported, used and recycled. This turnover is essential for life. Proteins that are damaged or no longer needed are broken down, and their amino acids are used to synthesize new proteins. Lipids in membranes are constantly being replaced. Even DNA, though more stable than other molecules, is continuously repaired and, in some cells, replicated. This dynamic quality extends to metabolism. The concentrations of metabolites rise and fall in response to enzyme activity, which in turn responds to the concentrations of substrates, products, and regulatory molecules. The system is constantly adjusting, seeking balance, responding to perturbations. Homeostasis, the maintenance of stable internal conditions, is not a passive state, but an active process requiring continuous energy input. When energy is no longer available, homeostasis breaks down and the system moves toward equilibrium with its surroundings, a state synonymous with death. Life exists far from equilibrium. It maintains gradients, differences in concentration and charge across membranes, which would spontaneously dissipate if not actively maintained. The sodium-potassium pump, for example, uses ATP to pump sodium out of cells and potassium in, maintaining gradients essential for nerve signaling and other processes. The proton gradient across the mitochondrial membrane, created by the electron transport chain, drives ATP synthesis. These gradients represent stored energy, potential waiting to be harnessed. Thermodynamics, the study of energy and its transformations, provides a framework for understanding biochemistry. The first law of thermodynamics states that energy cannot be created or destroyed, only transformed. The second law states that entropy, a measure of disorder, tends to increase in isolated systems. Living organisms appear to defy the second law, creating order and complexity. However, they do so by increasing the entropy of their surroundings. The heat released by metabolism, the waste products excreted, and the carbon dioxide released into the atmosphere all contribute to increasing the entropy of the environment. Life is not an exception to thermodynamics, but a beautiful example of energy flow creating temporary pockets of order. The concept of free energy, introduced by Josiah Willard Gibbs, is central to biochemistry. Free energy is the energy available to do work. Chemical reactions occur spontaneously if they decrease free energy. Reactions that increase free energy do not occur spontaneously, but can be driven by coupling them to reactions that release more free energy than they consume. This is the principle underlying ATP hydrolysis. The energy released by breaking the phosphate bonds in ATP is used to drive reactions that would otherwise be energetically unfavorable. Biochemistry reveals that life is fundamentally a chemical phenomenon governed by the same principles that govern all chemistry, yet giving rise to something qualitatively different, something that grows, reproduces, responds, and evolves. The molecules of life are not alive in themselves. A protein in a test tube does not grow or reproduce. But when these molecules are organized into cells, with membranes separating inside from outside, with metabolic pathways interconnected and regulated, with genetic information directing synthesis and repair, something emerges that we recognize as living. Emergence is a key concept. The properties of a system are not simply the sum of the properties of its parts. Water molecules are not wet, but a collection of water molecules is. Neurons do not think, but a network of neurons produces consciousness. Similarly, biochemical pathways, when integrated into a cell, produce life. This does not diminish the importance of understanding the parts. Detailed knowledge of molecules and reactions is essential, but it reminds us that the whole is greater than the sum, that understanding life requires looking not only at individual components, but at how they interact and integrate. As we approach the end of our journey, let us reflect on what we have learned. We began with atoms and bonds, water and macromolecules. We explored the pathways by which cells extract energy from nutrients and use that energy to build the molecules they need. We saw how cells communicate through signals, how genetic information is expressed and regulated, and how all of these processes are coordinated and integrated. We have observed that biochemistry is universal, that the same fundamental chemistry operates in all living things, from the simplest bacteria to the most complex animals. We have seen that biochemistry is dynamic, that life is not a state but a process, a continuous flow of matter and energy. We have learned that biochemistry is regulated at many levels, ensuring that the right molecules are made at the right times and in the right amounts. We have discovered that biochemistry is interconnected, that metabolic pathways, signaling pathways, and gene regulatory networks are woven together into a seamless whole. And we have appreciated that biochemistry is the foundation of health and disease, that understanding it opens doors to medical treatments and biotechnological innovations. Biochemistry also connects us to the history of life on Earth. The molecules and pathways we have discussed are ancient, refined over billions of years of evolution. When you breathe, when you digest food, when your heart beats, you are participating in processes that have been unfolding since the earliest cells. The same enzymes that catalyze reactions in your cells catalyze similar reactions in your ancestors and in organisms that lived millions of years ago. Biochemistry is a thread that connects all life, past and present, into a single grand tapestry, and so, as we conclude, we return to where we began. In the quiet hours, when the world slows, and the rhythms of life settle into a gentle hum, there is a rhythm at work beneath the surface, the rhythm of molecules meeting and parting, of energy flowing and transforming, of information being expressed and regulated. This rhythm has been with you always, from your first breath to this moment. It will continue as long as you live, sustaining you, adapting to your needs, maintaining the delicate balance that is life. Even now, as you rest, your cells are engaged in their quiet work. Glucose is being broken down, ATP is being synthesized, proteins are being folded, membranes are maintaining their integrity, genes are being transcribed, signals are being transmitted. All of this happens without conscious thought, guided by the principles of chemistry and shaped by billions of years of evolution. You are a living system, a flowing network of biochemical processes, a temporary but remarkable organization of matter and energy. Let this understanding settle gently. You are not separate from the chemistry we have explored. You are it. Every thought, every movement, every sensation arises from the coordinated activity of countless molecules. The chemistry of life is your chemistry, and in understanding it, even partially, you glimpse something profound about what it means to be alive. The pathways we have traced, From glycolysis to the citric acid cycle, from transcription to translation, from signal reception to gene expression are not abstract concepts. They are real, physical processes occurring inside you right now. The enzymes are folding and catalyzing. The ATP is being used and regenerated. The gradients are being maintained. The balance is being preserved. All of it working together in harmony, sustaining the miracle of your existence. Biochemistry teaches us humility and wonder. Humility because it reveals that we are not separate from nature, but part of it, subject to the same chemical laws as every other organism. Wonder because it shows how complexity arises from simplicity, how the intricate beauty of life emerges from the interactions of molecules following simple rules. There is elegance in this, a kind of poetry written in the language of chemistry. As you continue through your days, know that this molecular world continues its work. When you eat, your digestive system breaks down food into its component molecules, and those molecules enter the pathways of metabolism. When you exercise, your muscles increase their rate of ATP production, calling upon stored glycogen and fat. When you learn something new, neurons in your brain form new connections, synthesizing new proteins to strengthen synapses. When you heal from an injury, cells divide and migrate, synthesizing new tissue guided by signals and gene expression. Biochemistry is not confined to laboratories or textbooks. It is the foundation of your existence, the invisible machinery that makes life possible. And though much has been learned, much remains unknown. New discoveries continue to reshape our understanding, revealing deeper layers of complexity and integration. The study of biochemistry is an ongoing exploration, a conversation with nature that has been proceeding for centuries and will continue for centuries more. May this knowledge bring you a sense of connection to the molecular world within you, to the organisms that share this planet, to the history of life stretching back billions of years. May it inspire curiosity about the processes that sustain you and about the countless details still waiting to be discovered. And may it offer comfort in the knowledge that, even in moments of stillness, you are part of something dynamic, something ancient, something profoundly alive. The chemistry of life continues, always and without pause. It flows through you like a river, connecting past to present, molecule to organism, simplicity to complexity. You are carried by this flow, sustained by it, shaped by it. And in the quiet moments, when the world slows and you turn your attention inward, you can sense it, not with your conscious mind, but with the deep awareness that comes from being a living system, part of the great biochemical dance that is life itself. Rest now in this understanding. The molecules know their work, the pathways are established, the balance is maintained. You are held by chemistry, by the same forces that have sustained life for billions of years, and will continue to do so. Trust in this, in the wisdom encoded in molecules, in the reliability of biochemical processes, in the harmony of the living system that is you. And so our journey through biochemistry comes to its gentle close, not because there is nothing more to learn, but because we have traced the major currents, observed the essential patterns, and glimpsed the unity that underlies it all. The details are infinite, but the principles are clear. Life is chemistry in motion, molecules organized and regulated, energy flowing and transforming, information stored and expressed. It is complex yet comprehensible, ancient yet continuing, microscopic yet universal. May you carry this understanding with you, a quiet awareness of the molecular world within, an appreciation for the elegance of biochemistry, and a sense of wonder at the fact that such processes, so intricate and so precise, have given rise to consciousness itself, allowing you to contemplate the very chemistry that makes your existence possible. This is the gift of biochemistry, not just knowledge, but perspective, a way of seeing life that is both scientific and profound. The river flows on, the dance continues, and you, a living embodiment of biochemical principles, rest in the certainty that all is as it has been and will be, carried by processes older than mountains, sustained by chemistry as reliable as the turning of the earth. In this, there is peace.